On to immunity. So now, since the time of COVID, everybody speaks about vaccine, right? Everybody speaks about immunity. You get hundreds and thousands of forwards on WhatsApp, on all social media, asking you to eat this, that, right? You drink this juice, but none of them are ever tasty, right? None of these remedies or these suggestions, they always bring in bottle guard or bitter guard. They never tell us to eat all this pastries, sweets, and then say you'll get immunity. No, that rarely happens, right? Whatever food they suggest is very, some things which bore us, but they say you have to eat that, then you will get immunity. Fine. Immunity. So do we have immunity? Yes. We have two kinds of immunity. What is immunity? That is the inbuilt mechanism of a body to fight against disease causing infections is called as immunity, right? So every day in and day out, we are interacting or we are coming across so many different viruses, bacteria, protozoans in our environment. They are entering into our body either through the eyes or nose or mouth. So through various means, so they are through cuts, wounds, so through all of this they are entering into our body. Still we are not falling sick every single day, right? So the ability of the human body to combat, to fight against diseases, to fight against the incoming pathogens, that is called as immunity. So under immunity we have two kinds of immunity. One is innate immunity and the other one is acquired immunity. Under immunity, we have two types. One is innate immunity and the other one is acquired immunity. So what is innate immunity? Innate immunity is the one which we have, which is present in us right from the time of birth, isn't it? So even from the time of birth, we have immunity within our body. That is called as innate immunity. So it is a non-specific type of defense. So it is not specific for any virus or bacteria. Overall, we have an immune system in our body which protects us from other invading pathogens. That is innate immunity. Then there are four types of barriers which protect us from getting any infection. First one are the physical barriers. So what are physical barriers? Skin. Skin is a very good physical barrier, isn't it? So that is why skin is it doesn't let any other pathogens to enter into a body, right? So what is the pH of skin? Is it acidic or basic? Tell me, let's see. Okay, then again we have the mucus coating on the respiratory, gastrointestinal, everything. So again your nose, your respiratory organs, they all secrete mucus, right? So many times you must have thought, why is it that our nose secretes mucus? Because ultimately we discharge it, we throw it out of our body. But this mucus is very essential to trap the incoming microorganisms. So air is one medium through which these microbes can enter into a body. So as and how we breathe every day through our mouth or through our nose, the air enters inside the nose with lot of microorganisms, with lot of pathogens which can cause disease in us. So this mucus membrane which is present inside our nose and in our respiratory system, that traps these microorganisms and it kills them. So that is the function. This is also another barrier. Okay, skin is one important barrier. Okay, another mucous membrane. So like this we have various. Again the eyes also, eyes secrete tears, isn't it? So what happens, suppose you go out and some dust falls into your eyes, immediately without you asking the eyes to do anything, voluntarily or involuntarily the eyes will start secreting tears or water, right? So that is with the intention of throwing out that particle which is trying to enter into your eyes, isn't it? So these are the various physical barriers. Then we have physiological barriers. So in addition to that, our stomach and our mouth has an acidic pH. So this acidic pH also adds to killing the invading pathogens or microbes. So when we eat something unhygienic, say we go to a street side store and we have this golgappas, pani puri, right? Those are the most tempting items for us, isn't it? Especially now during December time, you can find in Delhi, there'll be a lot of these momo stalls. So on the street side, just you pass by those stalls, it will give such a good smell, you cannot resist. So you'll be tempted to go buy a few momos, but we know how food is made on streets, stalls on the street. So in very unhygienic conditions. So we buy that, and we, on a day-to-day basis, we are putting a lot of microorganisms into our body, isn't it? So the acidic pH of the mouth, that is the saliva, that also again plays a role in killing the pathogens which are entering into our body. And again, the stomach also has a acidic pH. Okay, there are, even in the stomach, the condition is acidic. That is because any pathogens which come into will be killed because of the acidic nature. Then again, we have cellular barriers like WBC, that is the leukocytes, right? Leukocytes or the white blood corpuscles. They also take part in, they are also a kind of cellular barriers. And we have cytokine barriers. So what are cytokines? See, when there is a cell which gets infected by a virus, so some virus is entered and it infects the cell. So this cell will produce components called as cytokines, okay, which protects other cells. So there are some other cells over here. These cytokines protect the other cells from the infection by the same virus. This is called as cytokine. Did you get it? So suppose there is one cell, there is a cell which gets infected by a virus, and this cell will produce components called as cytokine. Okay, so there will be other cells in the vicinity, isn't it? So this cytokine will go and protect all the other cells nearby, so that those cells also don't get affected by the virus. Got it? So these are various barriers, which these are all present right from the time of our birth. This is called as innate immunity. So first, under immunity we have innate immunity and acquired immunity. Innate immunity is the one which is present in us right from the time of birth, right? That is why it is also said that even the milk which a child gets from the mother, right, that is very, very essential in developing the immunity in the child. That is why it is, earlier days people did not have much knowledge about that. They just thought that it is the same as any cow's milk. So why should we go ahead, and further with many women working and all, they could not afford time to feed their babies. But nowadays there is a very good awareness among young moms that about the importance of breastfeeding the child, and especially the first milk after the delivery of the child, that is called as colostrum. I'm sure you have heard of colostrum, right? So it is a thick yellow color. It will be very thick yellow color milk. So immediately after childbirth, the first milk is called as colostrum. So usually it is a very, there are a lot of taboo associated with this. So it is said that in villages and rural areas and all, people did not have very, good knowledge about what is the importance of colostrum. So they thought it is it is something which is bad for a child. So what they would do is they would encourage the mother not to feed the first milk. They would discard it. And then afterwards, when the milk becomes white and thin, they used to feed only that milk to the child. But later, they, doctors started suggesting that it is this colostrum, which is very, very important. Colostrum has a lot of antibodies. I'll be discussing what is antibodies. Okay, colostrum is a rich source of antibodies, which enables in developing the immune system, very good immune system in the baby. So this colostrum should not be discarded at all. And the very first time, this colostrum has to be fed for the newborn child. Okay. Fine. Next, acquired immunity. What is acquired immunity? Acquired immunity which we develop after the birth, right? That is called as acquired immunity. So innate immunity is not specific to any particular virus or bacteria, nothing like that. It is an overall general immunity, whereas acquired immunity is the immunity which we acquire later after birth. Say first time we get some disease, say a common cold or a cough or a fever or chicken pox or smallpox, any of these. So the very first time our body gets exposed to that disease and that disease causing pathogen, then after that time it will succumb to the disease. Say for the very first time you get a common cold or flu. Okay, so the organism will come in contact, this pathogen will come in contact with the human body for the very first time, and that is why you are getting the disease. But after once you get it, now the body starts producing antibodies against that. Okay, antibodies are proteins which are specific to particular antigen. Say this is the microorganisms which are causing the disease. They are called as the pathogens or the antigens. Okay, so in response to that, what our body produces, that is called as antibody. Did you understand? So what is a pathogen? A pathogen can be a virus or a bacteria or a protozoan, okay, or anything which causes a disease in us. This is called as a pathogen. A disease causing organism is called as pathogen. And this is a virus. This virus will have certain structure, it will have certain proteins on its surface. Say let us take the example of our famous coronavirus. Okay, the structure is like this, isn't it? It has structure proteins. Okay, so first time, I think many of you all would have got COVID already. I don't wish that for you. Okay, but at least many of you all would have got it. So what happens to your body when you get COVID? I'll tell you. See, say this is the COVID virus, coronavirus, not COVID virus. COVID is coronaviral disease. This is the coronavirus. It is like this, right? It is like this. And when this when lockdown was happening, many people got crazy ideas. They started making cakes in the shape of coronavirus, okay, food articles in the shape of coronavirus and consuming it. What happiness do you derive out of that? Make food which makes you happy, isn't it? So make it in the shape of a heart or make it in the shape of a flower. All these things make you happy and consume it, fine. But why make it in the shape of coronavirus and consume it? What kind of happiness is this? Okay, fine. So say this is coronavirus, it is entering into your body. So first time when you get COVID, first time when you got corona infection, your body will start generating antibodies in response to that. So in response to this particular coronavirus, it will start developing antibodies. Say after few more days, okay, so this is after few more days, once again the another coronavirus tries to enter into your body and cause disease in you. So now your body has already produced antibodies which will go and fight against that antigen and it will kill it. So these antibodies will go and fight against that and it will kill this coronavirus so that it cannot create a disease in you once again. Okay, so did you understand what's the meaning of pathogen? Pathogen means any disease causing organism. Antigen means this specific. Okay, so the specific virus or bacteria, that is called as antigen, and the entry of an antigen into a body produces a response. Say for example, you can consider what happens in our borders. Say we have the Indo-China border or the Indo-Pakistan border. So when so suppose this is India border and this is the Pakistan border. So from Pakistan, if people are trying to enter over here, our soldiers will be here and they will catch hold of them and kill them, isn't it? Similarly, the antibodies also function. So first time when you get infected, the body is not prepared for that virus because it is a new virus, isn't it? So throughout your life, you would have come across many other pathogens which has got infected you, but this was something new. So now once you got infected, that time the body starts producing antibodies. Then after some time, once again when you get that same virus attacking you, now the antibodies are very well prepared. They will go and catch hold of that antigen and it will kill them so that you not get the disease once again. Got it? So in acquired antibody, it is pathogen specific. So innate immunity is overall general immunity. So immunity among most common things and protecting our body from external pathogens. Whereas acquired immunity is specific. That is as in how we get diseases after birth. So once we'll get cold, once we'll get some other chicken pox, smallpox. So like this, each time we get the disease, our body starts recognizing that and it creates antibodies and it has a memory. Our body immune system has a memory. So next time when the same pathogen comes and tries to create disease in us, it will combat it and it will kill it. Fine, so that the pathogen cannot create disease in us. So this kind of immunity is called as acquired immunity because it is pathogen specific. So under acquired immunity, we have B lymphocytes and T lymphocytes. Okay, so B lymphocytes and we have T lymphocytes. So it is the B lymphocytes which produce the antibodies. So whenever we get infected by a particular organism, our body generates, that is the B lymphocytes present in our body, they synthesize the antibodies. And it is the T lymphocytes which help the B lymphocytes to generate the antibodies. Got it? Okay. The T lymphocytes are also called as T cells. The B lymphocytes are the ones which generate the antibodies in us. Whenever we get an infection, it is the B lymphocytes which identify that and creates antibodies and keep it ready. So next time you get the infection from the same pathogen, it will go and combat. So it is the T lymphocytes which assist or which help the B lymphocytes to produce the antibodies. Because these antibodies are found in the blood, it is called as humoral immune response. So the immune response which is generated by the B lymphocytes is called humoral immune response, and the immunity which is generated by the T lymphocytes is called as cell-mediated immune response. Got it? Why is it called humoral? Because the antibodies are antibodies are found in the blood, right? Anything related to blood is called as, it starts with hum, H-U-M. So did you understand what is B lymphocytes and what is T lymphocytes? B lymphocytes are responsible for humoral immunity and the T lymphocytes are responsible for cell-mediated immunity. Then we have active and passive immunity. We have active immunity and passive immunity. We have active immunity, we have passive immunity. What is active immunity? So I told you, whenever we get exposed to any microorganism, say a virus or a bacteria enters into our body and our body starts producing antibodies against that, that is called as active immunity. Whereas sometimes there will be a case where we will be injected with ready-made antibodies, say in the case of tetanus injection, TT, right? So we discussed about tetanus injection in a previous class also. So what happens, you have a fall or you have a cut and you come in contact with soil, and there is a possibility that the the virus or the bacteria or the virus, all this would have entered into your body, that is the soil microorganisms would have entered into your body. At this time, because they are very lethal, so this time you cannot expect the body to create antibodies and then fight against that. Immediately you need to kill those microorganisms. So what you will do, you will take a tetanus injection. So these are injections which contain antibodies which are specific to those soil microorganism. Okay, so which is the organism which causes tetanus? Clostridium tetani, isn't it? So we have specific antibodies made against this particular organism, and that will be administered to the person in the form of an injection, in the form of tetanus injection. This is called as passive immunity. Same is the case with snake bite. So what happens when a snake bites? You cannot wait for the body to understand what is that a pathogen which has come inside, analyze that, then produce antibodies against that. By then you will be dead, isn't it? So immediately we need to give certain we need to combat that poison which has entered into that. For that we will give injections, isn't it? That is the pre-made antibodies. Okay, so that is, these are ready-made antibodies which are injected into the body to fight against that poison which has entered into us through snake bite. So this is called as passive immunity. Did you understand active immunity and passive immunity? Fine. Now moving on to autoimmunity. So sometimes what happens is our immune system recognizes our very own cells and tissues as something foreign. Okay, so our immune system identifies our very own cells which are present inside our body as a foreign particle or a pathogen, and it starts attacking that, leading to a disease. This is called as autoimmune disorders or autoimmune diseases. A very good example for autoimmune disease is rheumatoid arthritis. Rheumatoid arthritis. So this happens when our own immune system considers our cells and tissues as foreign, or considers them as a pathogen which is invading into our body, and it starts destroying it. Okay, this condition is called as autoimmune disease. Then now I'll just tell you briefly about the immune system which is present in our body. So what are the various immune organs and what are their functions? I'll show you a picture for that. So, okay. So our immune system consists of lymphoid organs, tissues, cells, antibodies. All of these constitute the immune system. So lymphoid organs, so the organs where the origin and the maturation of the lymphocytes occur. Okay, that is called as, what are lymphocytes? Lymphocytes are nothing but WBCs, right? So the place where the origin and the maturation of the WBCs or the lymphoids happen, okay, lymphocytes happen, that is called as the lymphoid organs. So there are two important lymphoid organs, that is bone marrow and thymus. Bone marrow and thymus. So this is the bone marrow. So how do you, if people, if many of you are consuming non-vegetarian food, you can find that inside that bone there will be a hollow, isn't it? Have you noticed that? That is the bone marrow, right? So that is the bone marrow, and that is the place where origin of the lymphocytes happen, birth of the lymphocytes happen. So under lymphoid organs we have primary organs like the bone marrow. This is the bone marrow, and the thymus. Thymus is a lobe-shaped organ which is situated close to the heart. Okay. So it is in the bone marrow and the thymus that the lymphocytes mature into antigen specific antibodies. Okay, so there they will have their origin, they will have their maturation, and then they are migrate. Okay, after maturation they migrate into the secondary lymphoid organs like the spleen. This is spleen, right? This is spleen. This is also a lymphoid organ. This is a secondary lymphoid organ. Bone marrow and thymus are the primary lymphoid organs, and spleen, okay, then lymph nodes. See, these are the lymph nodes. These are present in between tissues. So lymph nodes, spleen, and even the tonsils. Okay, here you have tonsils. That is also a secondary lymphoid organ. So what happens? First, the origin and maturation of the lymphocytes, be it the B lymphocytes or the T lymphocytes, happen in the primary lymphoid organs such as the thymus and the bone marrow. After maturation, they are migrated into the secondary lymphoid organs. Okay. Then so here you can see again, this is the tonsils. Okay, thymus, which is a primary lymphoid organ, bone marrow, so the hollow inside the bone, that is called as the bone marrow. Then you have spleen, you have lymph nodes. So many a times what happens in patients with severe organ damages, they will be suggested to go for a organ replacement surgery, isn't it? So one of their close family members will be identified who have the same blood group, who has the same, we have many things which we have to match, like the histocompatibility complex, all these things. Even after doing all that and the organ is taken out from the other donor, and it is put into the person who has the organ failure, that is a patient. Even in those cases, you can find that the organ which has been transplanted will be rejected immediately. So it is the cell-mediated immunity, that is the T lymphocytes, which are responsible for this act. Okay, so that is why in case of organ transplantation, the patient will be given immunosuppressant drugs for a very long time, so that the organ can adapt to the body and the body will start accepting it instead of rejecting it straight away. Okay. So this is, next we shall move on to chromosomal banding. Allergies, you know what is allergy, right? The sudden exaggerated response of the immune system to certain antigens present in the environment, that is called as allergy. So many people will be allergic to dust, many people are allergic to pollen grains, especially people from other states. Many of them have complained when they come to Bangalore. So in other people living in other states who do not, where the place do not have huge number of flowering trees, when they migrate to Bangalore, because of the huge quantity of pollen grains that is present in the air in Bangalore, they become allergic to that, isn't it? Similarly, there are many different allergies. So again, the doctors will prescribe antihistamine tablets like Allegra and all for that. That is also a immune response only. Allergy is also a exaggerated immune response to the antigens present in the environment. Okay. Now we shall discuss chromosomal analysis or chromosomal banding. Chromosomal analysis. So we all know that chromosomes are best visible during the metaphase stage of the cell division. So what we do in chromosomal analysis is we take actively dividing cells such as the bone marrow cells. So I told you already, bone marrow. What is bone marrow? That is where the origin of the WBCs happen, isn't it? So it is the bone marrow where we can find lot number of actively dividing cells. So these cells are taken, okay, actively dividing cells are taken, and a protein extract called phytohemagglutinin is added to the cell culture. This is very important, underline this, phytohemagglutinin. It's a lengthy word, but it is important. Please note it down. So phytohemagglutinin, which is a protein extract from kidney beans, it is found that this phytohemagglutinin induces DNA synthesis. So what we will do is we'll extract the cells, okay, preferably actively dividing cells from the bone marrow or any other place where we can find the actively dividing cells. And in that culture, we will add phytohemagglutinin, which induces DNA synthesis. In addition to this, we will also add colchicine. So colchicine is a chemical which prevents the hybridization of spindle fibers. Now, when the cell is actively dividing, first it will be in prophase. Then it will move to anaphase. Prophase, metaphase. Then it will move to metaphase. Then it will move to anaphase. Then telophase. Then cytokinesis, etc. Isn't it? So when we have to study the cell, the best phase to study the cells is metaphase. But by the time we study, it would have already within seconds, even within milliseconds, it will move to the next stage, that is anaphase. So now what we have to do is, if we want to study the chromosomes and this metaphase stage itself, we have to arrest the further cell division. Okay, we should stop the cell from moving to the next phase, that is anaphase. So now what we will do, we will add this chemical called as colchicine. So colchicine arrests the cell division at that stage itself. So the cell will not continue to the next stage. So the cell will stay at metaphase stage itself so that we can study it properly. So that is why we are adding colchicine. So what is the purpose of adding phytohemagglutinin? It is found to induce DNA synthesis. That is why we are adding phytohemagglutinin. And then we are adding colchicine to arrest the cell division, to arrest the cell at metaphase stage itself, so that we can study it thoroughly. Then after this, we will, the cells are fixed, okay, and then the bands are observed. So there are 4 types of staining procedures. First one is the Q banding or the quinacrine banding. I'll show you pictures for that. Q banding or the, see, this is how the sample is made for the banding. Okay, so the sample is taken and the cells are grown in the laboratory conditions. Then you here you will disable the mitotic spindle. That is, here in this step you will add colchicine. Okay, here you are stimulating the cells to divide. Okay, then after that you are arresting, once they start division, you are arresting the cell division at the metaphase stage itself by addition of colchicine. Then you will study the chromosomes over there. Fine. So Q banding. So there are 4 types of staining procedures. Okay, 4 types of staining procedures. First one is the Q banding or the quinacrine banding. Here the bands appear fluorescent under the fluorescent microscope, and the bands rich in AT. Okay, the bands are rich in AT compacted by proteins. So here you can see when you do this quinacrine banding, okay, or the staining procedure, under a fluorescent microscope you can find that the chromosomes will appear as, they will start fluorescing. You can see them, isn't it? Here you can see it as fluorescent yellow or fluorescent green. This is how they will fluoresce and you can observe it. Okay, so this is a metaphase. So we stopped the cell division as metaphase plate, we stained it, and we are observing this under the microscope, under a fluorescent microscope, where these bands, because of the fluorescent banding technique, that is a Q banding or the quinacrine fluorescent banding technique, we utilized to stain these chromosomes, and we are observing it like this. Okay, next one, G banding or GMSA banding. Okay, here in this G banding or GMSA banding, the chromosomes will appear as darkly stained regions. Okay, so the darkly stained regions are the AT rich region. So I told you what is AT, right? Adenine, guanine, cytosine and thymine. They are the 4 nucleotides, isn't it? So adenine and thymine will base pair. So those regions which are AT rich will appear as dark blue color. Okay. Next, moving on to C banding or centromeric banding. So this is again a staining technique, okay, where only the centromeres and the satellite region will get darkly stained. Here you can see this is the centromere, these are the arms, P arm and the Q arm. Here you can see only the centromere portion will appear darkly stained. This is the C banding. Got it? This is the centromere. Again, here you can see only the centromere is deeply stained. Or the satellite region. This is the satellite region. This is again the centromere. Here it is a satellite region. So in this C banding technique, only the centromeres and the satellites will appear as darkly stained regions. Next, moving on to R banding or reverse banding. Okay, so you might ask me, what is a satellite? So this is called as sometimes there will be a secondary constriction. Okay, here there is one constriction at the place of the centromere. Okay, in addition to that, sometimes there will be a secondary constriction, and that remaining part after that secondary constriction, that is called as a satellite region. Got it? That's why I took this picture to show you. Then we have the R banding or the reverse banding. Then we also, here this banding develops because of the differential response of the chromosomal proteins to denaturation. And NOR banding. So another staining technique is NOR banding. So here you can find that these satellites, which are also called as the nucleolar organizing region. So what is the full form of NOR banding is nucleolar organizing region. In short, we call it as satellite regions. So it is these satellites which get stained. Okay. So here in this NOR banding, you can find that these satellites, which are also called as nucleolar organizing region, it is this region which appear as darkly stained. Fine. So it's already time up now. I had sent some more material for today's class, that is genetic screening, genetic counseling, genetic imprints in human disease. However, this chapter itself got extended until now, and we could not even start off with the new chapters. Anyway, do not worry. After your winter break, we shall discuss those topics. Fine. If you have any queries, now you can send it. You can message me. I will discuss them and we shall wind up.